So for this part, muscles and muscle tissue, we are going to be getting into more or less the different types of muscles that we have in the body, how they compare to one another, and how they contribute to the overall force and also speed of contraction. So in a muscle contraction, all the same principles apply to single fibers as they do to the entire muscle cell. So whenever you're looking at the contraction sequence, we're explaining what's happening at the sarcomeres. But remember, the sarcomeres are inside the myofibrils, which are inside of the fibers, and those are the cells, and they're contracting. But when we contract those fibers and we shorten those fibers, we're contracting and shortening the entire muscle. We started down at the microscopic level, but make sure you kind of blow it up and go all the way out to the largest level. Contraction is when we have muscle tension and we are pulling or exerting a force on an object. We do think about contracting and shortening muscles, but that's not always the truth. In a contraction, we may or may not shorten the muscle. We have something called isometric contraction and then isotonic contraction. Isometric means same length. That prefix iso means same. Metric means length. Same length contraction is where we don't have any shortening, but we're still applying a force. So the best example of this is whenever you're pushing on something. If you try to move one of the tables in the room, you usually push on them, and that force doesn't really change the length of your muscle, but you're still applying a force, and that would be the isometric. Isotonic, that is when the muscle does shorten, and we do move the load that way. So isometric does not shorten, but isotonic does shorten. We can also change the force and the duration of the contraction by changing the stimulus. And remember, this stimulus for skeletal muscle is coming from your nervous system. It always has to come from the nervous system. The stimulus can change its frequency, which is how often the stimulus is coming in and telling the muscle to contract. And it can also change intensities, which is how strong it's communicating with the muscle fiber. Each muscle is served by one motor nerve, and we explain what that is in the next chapter as far as a motor nerve, but we know that these nerves are communicating. The motor nerves, we said, branch out into axons, and we have hundreds of these motor neurons and the motor cells that we're going to be identifying. The motor unit is what we're going to identify next. This is one nerve and then one of the fibers that it's connected to, and that communication for the neuromuscular junction. So a motor unit, here's your definition. It's a motor neuron and all of the muscle fibers it supplies. Motor neuron and all of the muscle fibers that it supplies. It could connect to 4 muscle fibers, or it could connect to hundreds of muscle fibers. So motor units can be small or they can be large. And I think I kind of introduced this and got you thinking about this not Tuesday, but I think it was Thursday last week. And I asked you guys, when you contract muscles, do you always contract them with the same amount of force? And you guys said, no. Sometimes we use a little bit of contraction, sometimes we use a lot. If it's small, we don't need to use every single fiber because that would be a complete waste of energy. So whenever we start contracting a muscle, we always start with the smallest motor units first, and then we eventually work our way up to the larger motor units. The smaller ones are for fine motor skills, like for your fingers and picking up light things, and then the heavier ones are going to be contracting more of the entire muscle, more of a force for the contraction. So for this figure, it's showing you 2 motor units. This is a section through the spinal cord, and then this would be a spinal nerve that branches off the spinal cord. And in the spinal nerve, that's where we're going to be seeing some of the neurons that we're going to be learning about in Chapter 11. Motor unit number 1, you can see that we go out and say connect to a couple of muscle fibers. Motor unit 2, we connect to maybe 3 fibers. If we compare the two, motor unit 1, if we connect to these muscle fibers, it's not going to be as forceful of a contraction as we are in motor unit 2. More fibers means a more forceful contraction. Does that make sense to everybody? More fibers means more force and a stronger type of contraction. So that is the motor unit and how it controls the force of contraction. Before we talk about muscle twitch, I want to kind of give you an example of a motor unit, something else that you do every day. Students always ask me, how do we know which motor units to use? Do we use 1? Do we use 2? Do we use 5? What do we use? That's something that you start to learn as a kid. Your brain actually starts to program and memorize which motor units we need to pick up. So whenever you look at an object, we assess it, right? We look at it and we say, that's small. It's probably not going to be very heavy. Or we say, oh, that's big. That's probably going to be very heavy. And by looking at the object and by figuring out about its size, that's how your brain says activate X number of motor units. So if you've ever gone over to a big box and you pick it up and it's not as heavy as you thought it was going to be and you kind of jerk, that's because your brain said the box is heavy. Activate all of these motor units, and we forcefully contracted when we didn't really need to. We do the same thing sometimes whenever you pick the milk up out of the refrigerator. If your family's like mine, they put the empty container back in the refrigerator. So then you go to pick up the container and you're just, what happened to the milk? You know, you forcefully contract and you sling your arm and there's nothing. Yeah, you didn't need all of those forceful contractions. So we use those motor units just by assessing the size, and that's something that we learn to do way early on by looking at the size and figuring out which ones we need to do. Okay, muscle twitch. Muscle twitch is the simplest contraction that we can have from a motor or from a muscle fiber's response to a single potential from a motor neuron. So that's kind of the complicated definition. Let me break it down for you. This is one motor neuron telling the muscle fiber to contract. It contracts, it relaxes, it's just one single response. So one single response is very quick, and then it relaxes, and we can actually measure the sequence of these muscle twitches on what we call the myogram, where we trace the electrical activities in the muscle fibers. There's 3 phases of a muscle twitch, which is one single response from one single neuron. The latent period is the period where the muscle fiber is experiencing excitation, contraction, coupling. Excitation, contraction, coupling is where we're getting to the point of releasing calcium, but we haven't actually created tension and contraction or changing in the length of the muscle. As far as the period of contraction, that is where the calcium is binding to the troponin and we're moving the tropomyosin and we form the cross bridges. That is when we have the tension and we're applying the force. Last period of relaxation, that is when we take the calcium off of troponin and we put it back into the sarcoplasmic reticulum. We said that we recycle it, so we already know that. And the tension will then go back out to its original length. So as you look at this list, I'm looking at it now, it should have numbers there because they're in order. Latent period is first, period of contraction is second, and period of relaxation is third. So if you want to write that down in your notes, they are in a sequence. So in a typical muscle twitch, the latent period here, right after we receive a stimulus from the nervous system, is the shortest period. That's when we're experiencing EC coupling, when we're spreading the action potential down through the sarcolemma, down into the T-tubules, and we're releasing the calcium. Here, period of contraction, that's when the calcium binds to the troponin, and the troponin is pulling on the tropomyosin. We form the cross bridges, and we're actually contracting the muscle. And then last, we have the period of relaxation, and that's where the calcium is being pulled off the troponin, put back into the sarcoplasmic reticulum to recycle. Not all of your muscles have the same length and time for their response to a single stimulus from the nervous system. Different lengths and different durations in these muscle fibers depends on the metabolic properties inside of the muscle cells. We have different metabolic properties, different chemical reactions going on. We also have different enzymes. And a couple of examples, the eye muscles that are responsible for moving your eye, they contract very, very quickly. Whereas we have what we call the fleshy muscles, which are like in the calf area, those are going to contract more of a slow and a long endurance in time frame. So in this example, we've got something called the extraocular muscle. That is a muscle that's around the eye, and you can kind of break it down by the name. Ocular is the eye. And if we have a single stimulus going to the extraocular muscle, we get a really rapid contraction and relaxation. The sequence on the myogram is very, very quick. If we compare that to two of the muscles that we have in the calf area, gastrocnemius, soleus, both of those are on the back of the calf. If you're not learning muscles yet, you're going to get there, but that's where they are. They're right back there on the calf area. The gastrocnemius, if we have one single stimulus, it's kind of moderate in its length as far as contraction to relaxation. And the soleus is really long, slow contraction and relaxation. So again, as far as the length of the response to a single nerve impulse, it depends on metabolic properties, which is how fast we're making ATP, how fast we break down glucose, and we're going to talk about all of that, and also some enzymes that are responsible for the contraction sequence. Yes?
What is it when you're just sitting still or sitting down, your leg is like your leg muscle keeps twitching?
We're going to talk about that. We're going to talk about that. Okay. Usually whenever you have those twitches, it's because of an ion imbalance. So it's kind of like a muscle cramp. Whenever you exercise for a long period of time and you'll get your muscles stay contracted, they'll tell you to eat a banana. And that banana is really high in potassium. And we know that potassium is for the relaxation part at the end of the contraction. So usually when you have those, it's because you're low in potassium or some type of ion that you need. Mm-hmm. So eat a banana next time you have those.
Could it be like anemia related?
Sometimes it is because of the iron and the oxygen that's going to the muscle. But, really the short term is from the potassium. Mm-hmm. Sometimes it is the iron. Mm-hmm. Okay, good question. We'll talk about all that later on at the end of this lecture too. For muscles in the response, the stimulus we can change. We've already said in two ways. We can change the frequency of the stimulus, which is how often the neuron is telling your muscle fiber to contract, and that's what we're going to talk about now. So a single stimulus will result from a single contractile response, which is what we call the muscle twitch. Single stimulus, single response. But if we have multiple stimuli coming in back to back, that could result in what we call wave or temporal summation. This is where we have multiple stimuli, rapid succession, which means one stimulus very quick right after another, back to back in a sequence. If we send a stimulus very rapidly to this muscle to contract before we completely relax, that is going to increase the force. Okay, some of you may not exercise now, but you may have at one point exercised, and you may have lifted weights. So when you lift weights, do you curl the weights and then just flop your arms completely down? No, you keep them a little contracted, right? And then you do another curl. And then you come down and you do another curl. And then you get to about 10, and then your arms are burning, right? Which is what you want. You want the burn. The reason that that's happening is we don't completely relax the muscle. Your brain is saying, contract, relax a little, but then contract again, relax a little, contract again. And with each contraction, you're generating more force because we're activating more fibers. The more fibers we get, the more burn we get. So that keeps those muscles going in that exercise state. But we do the same thing even in different day-to-day activities. So at the point that we're stimulating another contraction before we completely relax the muscle, we have some more calcium being released, which means more muscle fibers are going to be contracted by these cross bridges, and we'll have this more force or increased force with each stimulus. We have two types of wave summation, one that's called unfused or incomplete tetanus, and then we're going to talk about next, which is fused or complete tetanus. If we have unfused or incomplete tetanus, this is when we have short periods of relaxation. Not complete relaxation, but partial relaxation. So this is when I was saying you're exercising. You curl the muscle, or you curl the weights, you slightly relax, curl again, slightly relax. That's going to be an incomplete tetanus. By the way, tetanus, don't let that throw you off. Tetanus is not the same thing as tetanus that you need a shot for. We're not talking about rusty metal here. Tetanus is a term that we use for a constant contracted state in muscle. Yes, tetanus bacteria does lead to constant contracted muscles, but this is your brain, in this state that we're talking about, this is your brain telling the muscles to stay in that constantly contracted state. So it's two completely different things. For the fused or complete tetanus, this is when we sustain a constant contracted state. There's not any periods of relaxation, not any partial, not any complete. We constantly contract the muscle. So this is when we pick an object up and we hold it. So some of you don't carry a backpack, you just carry books with you, or maybe you carry your backpack in your hand or a bag in your hand. When you pick up something and you hold it, that is a constantly contracted state. We're not letting go, we're not letting any partial contraction happen, and that would be the fused or complete tetanus. If we have prolonged muscle contraction, that could lead to what we call muscle fatigue. And muscle fatigue is when the muscle just absolutely breaks down. We don't have what we need for the contraction. Very rare, but we will talk about some situations where it could happen. So to see on a myogram, again, just to remind you what a single muscle twitch looks like. If we have a stimulus, which is the red arrow coming from the nervous system, is where that stimulus would come from, telling the muscle fiber to contract. We go through a short period of EC coupling, then we contract, then we relax, and we're done. That's a single twitch. If we show the picture of unfused or incomplete tetanus, we have a stimulus where we start to contract the muscle, we partially relax, but then we have another stimulus coming in and telling the muscle to relax again, and we contract, relax again, we have another stimulus, and we get this increased tension because we're recruiting more and more muscle fibers. So as you're looking at this picture, one thing that's kind of tricky, whenever we looked at the sequence of depolarization and repolarization, that sequence where we change the charge, there is the repolarization where we're closing closing sodium, opening potassium. If we have most of the potassium channels open and we're mostly repolarized, that's when we can respond to another stimulus. So we're going to learn a little bit more about that in nerves, but just for now, know that we've kind of gotten there to the point that we can respond to another stimulus. Fused or complete tetanus, this is where we said the frequency is going to increase so much that the muscle will never relax until the stimuli do not tell the muscle to relax anymore. So all of these little red arrows, again, are signals from the nervous system, telling that muscle, contract, 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 contract. You have to constantly tell that muscle to hold your book, and as soon as you tell the muscle to stop, you would release and let go, completely relax the muscle. So here's the take-home message about frequency from the nervous system. As we increase the frequency from the nervous system, we increase the force of the contraction. Increase the force of the contraction. Something else that we can change about the stimulus from the nervous system is the strength of the stimulus. And this is another kind of FYI that I think that your book should include, but hey, I didn't write the book, I'm not the author, didn't get paid for that, but I'm just going to tell you. Remember that the stimulus coming from the nervous system, how we communicated between the nerve and the nerve cell and the muscle cell, was with a neurotransmitter called acetylcholine. Remember that? So whenever I say strength, the strength that I'm saying is more or less acetylcholine being released. More acetylcholine means more changing in charge, which means a more forceful contraction, and then less acetylcholine, a less forceful contraction. We also use the strength in what we call recruitment, and that is in the number of motor units that we're going to be activating. So in a single response, if we have a low stimulus, that's what we call a subthreshold stimulus. We have to exceed the threshold, and we talked about that last time. Remember, we said we start off at -90, and then we have to get close to, it's around -50, and then eventually we start that action potential to go all the way up to +30. If we don't release enough acetylcholine, then the muscle will do nothing, and that's the subthreshold. Sub means under the threshold. Threshold stimulus, that's where we do exceed the threshold. We have enough acetylcholine being released, and we will respond with a twitch. We will contract, relax, the sequence will happen. Maximal stimulus is where we completely contract all of the motor units that we have inside of this muscle. Not very common, by the way. You never, ever, in a normal state, relax or contract every single motor unit in a muscle. So, have you ever heard of people being in these weird kind of situations or dangerous situations where they have to pick a car up off of someone, or they have to move a big boulder off of someone, and they think, oh my gosh, it was superhuman powers, I'm a superhero. No. It's just that in a normal state, we don't activate all of our motor units. But when we are scared, we do have the ability to really use some muscles and all of the motor units, and that would make that force so strong that we could pick up a car or pick up, whatever. In the case that we have maximal contraction, it's very tiring for the muscles because we use so much ATP and the muscles just completely collapse afterwards. And I used to show a video, I might try to find it and post it online if I still have it, of a guy that he was rock climbing and a boulder slipped and fell on top of him, and he pushed the boulder off. It was huge, hundreds of pounds. He pushed this boulder off of his chest. And that force of contraction, when he used all of his chest muscles, was so strong, he actually tore his muscles from his chest and actually broke some of the bones that he had in his chest. It's just crazy to think that your muscles contract that strong, but they can if we need them to. So as we increase strength, which is the amount of acetylcholine that we're releasing, we said that subthreshold, that is where we're not going to activate any of our motor units, so there's not going to be any tension, not going to contract at all. But whenever we exceed the threshold, that's when we start to contract, remember, first, the smaller motor units, which means just a few of the muscle fibers. And the muscle tension, a little bit, but not too forceful. The more acetylcholine, the stronger the strength, that is when we really start exceeding way up and activating more and more motor units so that we get a maximal force to our contraction. So that's how the nervous system is telling your muscles to contract, and we can change the force of contraction based on those two things. But we can also define something called muscle tone, and in muscle tone, this is your muscles staying in a slightly and constantly contracted state. This is due to spinal reflexes coming from the spinal cord, and it's constantly telling groups of motor units to kind of alternate and keep your muscles constantly contracted. The reason that we do this is to keep the muscles firm, healthy, and also ready to respond. It's kind of like a sprinter. They're down, ready, and anytime we have a stimulus coming in, we go ahead and jump in and respond. So we keep mentioning ATP as well, and I mentioned that some of the responses for your muscles are going to be very quick, some of them are slow, some of them kind of moderate. And one of the factors, as far as metabolic factors, that changes the length of our response for muscle contraction is how fast or where we're getting the supply for ATP. ATP is your energy all the way down to your muscles. This is how we contract muscle. We've seen it in the picture of the cross-bridge cycle. Remember, we had ATP come in and bond to the myosin head. We detach. Hydrolysis happens where we split it apart. Phosphate, all of that we've already learned. So ATP reminds you what it does. It detaches. I want you to underline this. It detaches the cross-bridge cycle. But if we didn't have ATP, the myosin head wouldn't move either. I told you guys that the myosin head already has a phosphate group, and it's just sitting there waiting for that tropomyosin to move out of the way so that we can grab onto the actin molecule. And that's exactly what we do. So ATP, it does trigger detachment, but if we don't have it, the myosin heads can't attach either. As we look at this, we first need to mention that you do have stored ATP in your muscle. You have a constant supply. It's kind of like in your little pantry. You have a little bit of ATP. But as we start using muscles, we become short. It says it's depleted, not completely deleted. It's still there a little bit, but we start to get low on ATP after about 4 to 6 seconds after using the muscle. Okay, in your book, it uses exercise a lot. And I like to use exercise because, that's when we really use our muscles. But that's not the only time that this is going to happen. If you park your car way over there in that parking lot and you start walking across the street, it's usually going to take you longer than 4 to 6 seconds to get here. You don't ever, if you're lucky to get a parking spot that you can get into the classroom in that short amount of time. So if you start walking 4 to 6 seconds, that's when your stored ATP starts to get low. When you're exercising, same thing, ATP starts to get low. When you're just hanging out at home and you're sweeping the floor or whatever you do at home, ATP starts to get low. That's any type of muscle, whether it's your arms, your legs, whatever. So after we start to get low on the stored ATP, that's when we will start 3 mechanisms to generate more ATP. And again, I want you to number these because they do happen in order. If you want to put number 1 here, number 2 here, and number 3 here. Right after we get short on the stored ATP, that is when we start the process of, don't panic, direct phosphorylation of ADP.
That's not in the list.
Okay, we'll write it down.
Okay.
I think they're just broke down.
It is broken
But
down.
they're not all in.
They're not in a list, but it's in your notes.
Okay.
Yeah.
I didn't see it at first.
So if you just wanted to kind of write it on the side that that's the list that you're going through, after we get the stored energy or get low on the stored energy, that's when we start to use these 3 processes. Okay, so as we all wrote that down, I want to kind of break down the first name. I don't want you to panic just looking at it because you're probably writing it down thinking, holy cow, what's that process going to be? Because I've already learned muscle contraction. What's fixing to happen now? It's not so bad once you break down the terms. You can probably recognize this, right? ADP. Okay, so that's diphosphate, which means we only need 1 more phosphate for TP, right? ATP. Phosphorylation, we've already heard that term also. Phosphorylation is when we add a phosphate group. So what we're going to do is add a phosphate group to ADP in order to make ATP. So not too bad once you break it down. And where we're going to get that phosphate is from something called creatine phosphate. So not so bad once you break it down. Think about it as why do we have that name to it? Anaerobic pathway is going to be called glycolysis sometimes, and it's also called lactic acid formation. And some of you may have had a class where you learned about glycolysis if you took maybe another biology class. We're not going to even get into all of that detail about glycolysis and breaking down this and that. We're going to make it very summarized and not very detailed. And then the same thing for aerobic respiration. It's just a little summary of the sequence. Okay, first, let's remember where we are. We're short on stored ATP, and now we're going into our first process of generating more ATP, which is direct phosphorylation of ADP by creatine phosphate. And this was, if you want to write your little number, number one after stored energy. So creatine phosphate has a phosphate, and it tells you that in its name. And it's a molecule that can donate that phosphate to ADP in order to form a full molecule of ATP. There is an enzyme called creatine kinase that is involved in the transformation and the movement of the phosphate group. But the thing about this process is it's not going to supply you with a whole load of ATP. It's a short amount of ATP, or a small amount of ATP, that will power the cells for about 15 more seconds. So we've walked across the campus 4 to 6 seconds, and now we've got 15 more seconds. You're probably still going to be walking, right? Which means we need to keep going and making more ATP. Before we get into the next step, I want to show you these pictures because it really summarizes how we have the process happening. Here's your creatine phosphate. Here's your ADP, and we have the creatine kinase, which is our enzyme for breaking off the phosphate and performing the chemical reaction. So what we get after we have the chemical reaction is not creatine phosphate anymore, it's just creatine, and now we have ATP, our full energy molecule. So what we get is one ATP molecule for one creatine phosphate molecule. That's going to be a lot of work, and it's not going to give us much energy. Again, it tells you 15 seconds. So you may know someone, or maybe you, I don't know, what you do whenever you exercise. There's creatine shakes that you can drink, or creatine supplements that you can take. I think they even have a pill format. And those creatines, they usually take either before their workout or during their workout, and that's to keep their energy going in the muscle to supply more energy. And here it is. We have a natural supply of energy, but if you're going to be at the gym for a long time, that's when those muscle bodybuilders are there for hours. They may take those supplements in order to get more energy. Number 2, let's complete our sequence. We've gone through stored phosphorylation with creatine phosphate, and now we're at anaerobic respiration. Anaerobic means without oxygen. Without oxygen. This is a chemical reaction that does not require oxygen. And sometimes we throw in the term glycolysis, the reason being we're going to be breaking apart a glucose molecule. Glyco is referencing the glucose, and then lysis is breaking apart of something. So we're breaking apart the glucose, the glucose, and in that glycolysis process, again, we do not require oxygen, and the end result is going to be 2 molecules that we call pyruvic acid. 2 pyruvic acids from every glucose molecule. And we also generate 2 ATPs through the chemical reaction. 2 ATP molecules. That's not very much either. Because remember, one single ATP molecule bonds to those myosin heads and stimulates the detachment. So if we only have 2, that's only 2 myosin heads that we can attach to. Again, we have millions of these chemical reactions happening, but it's not going to provide a long-term type of energy. Continuing on with the anaerobic pathway, what we do with the pyruvic acid molecules, after we've made them, after we break down the glucose, and we have these pyruvic acids, we usually want to send them into the mitochondria, which will then start the third process, aerobic respiration. However, if we don't have oxygen that we're going to use in the mitochondria, which is where we're going to convert the pyruvic acid, that is when, instead of pyruvic acid to more ATP, we're going to make pyruvic acid to lactic acid, and that's what we have here, lactic acid. Lactic acid is what builds up in your muscles and makes your muscles sore. Whenever you use them a lot, and that is what makes them tender and hard to use whenever you're walking and you're stiff. That's because you have more lactic acid in your muscle, or too much lactic acid in your muscle. So in the anaerobic pathway, we have our sugar molecules that perform or go through glycolysis, and by the way, that's happening in the cytosol of these muscle fibers. It's not in the mitochondria yet. Whenever we break down the glucose molecules, we get 2 pyruvic acid molecules, and these pyruvic acid molecules, if you're okay with writing in your book and kind of taking notes, you can write down that the pyruvic acid, that's where we want to go into the mitochondria, and then we use it for the next step. But if we don't have enough oxygen, because it's not going into the mitochondria and converted into ATP there, that's when we make lactic acid, which makes your muscles sore. And we also said that we get 2 ATP molecules, again, from every single glucose molecule. As far as how long this energy is going to last, 30 to 40 seconds, not very long. Again, we had 4 to 6, about 15, 30 to 40. You're probably still going to be walking from the parking lots and trying to get up the stairs at that point. Last, number 3. This is where we have aerobic respiration, and this process does happen in the mitochondria. This is where we're going to get most of the ATP. It actually makes 95% of the ATP that we are using for either at rest or light to moderate exercise, and even whenever you're doing aerobics. Haha. Have you ever wondered why aerobics is called aerobic exercise? It's because of the type of energy and the way that we're making ATP in the muscles. So we're taking the pyruvic acids that we've just made in the glycolysis in the anaerobic pathway, putting that into the mitochondria, but the other thing that we have to have is oxygen. We also have to have oxygen going into the mitochondria. As we are going through these chemical reactions, we are going to provide a lot of energy, a whole load of ATP molecules. And we're also going to provide enough energy so that we can use the muscles for long periods of time. It says 30 minutes or more because we really pump out ATP here. Something else that we're going to use, fatty acids. I don't know if you remember where those come from, but fatty acids come from lipids and fats, which is why we go to aerobics, right, ladies? We go to aerobics to burn fat. And those aerobic classes, you don't just walk in and they're like 5 minutes long. Because then you don't get to this stage where you're using fat to make energy, and that's what we want to do. We want to burn the fat to make energy and use our muscles. That's what we're doing in those exercise classes. So in the aerobic pathway, this is in the mitochondria. We've got the pyruvic acid coming from anaerobic pathway going into the mitochondria there. We also have to have oxygen going in, and we also said the fatty acids from the lipids. Something else to take note of that we have to have is amino acids, and you might want to write that down in your notes. Maybe next to the fatty acids, write down amino acids. Amino acids were the monomers for proteins. And if you have ever been on a really good exercise program, they always come with a diet plan. They don't just tell you exercise, but they tell you to eat well, right? And it's a lot of proteins that they tell you to take and eat, because if you're doing aerobic exercise, that means that you're going to be burning your proteins. You need proteins for so many different reasons. We know why. They're for chemical reactions, for enzymes. We even need them down to muscles, right? Your actin and your myosin, those were both proteins. So unless we replace that through a good healthy diet, which protein's going into the muscle, makes the muscle tone, and we're burning the fat that's over the muscle, that's going to be a good balance between the exercise. What we get is some carbon dioxide and water, and 32 ATP molecules. And when it says 32 ATP molecules, that is from one single pyruvic acid, some fatty acids, amino acids. We get 32. And there are millions of these chemical reactions happening inside of the mitochondria. So it's 32 times millions. That's how much we get from these chemical reactions in the mitochondria. So here's kind of the thing about the mitochondria. It was called the powerhouse because we said that that's where we make ATP, which is very true. But we also make ATP in other places, but that's just the primary place where we get most of the energy. And it also says that we get energy for hours out of this type of pathway. So in sports, or even in general kind of activities, but we use the examples of sports because that's when we really know that we're using muscles. Aerobic, endurance types of exercise, that is where we exercise, maybe running on a treadmill or swimming, long lengths of muscle contraction. If you're a runner and you run long distance, that's aerobic. If you're a swimmer, that's aerobic. If you just walk even, that is aerobic. Anaerobic is where we are doing more of the powerlifting and that kind of stuff and resistance type of training. If you want to write down anaerobic and resistance, that's where we're lifting weights. So, I'm not a personal trainer. I'm not a trainer at all. I like to exercise, I do, but I don't really know exactly what the trainers tell you to do. I just go to the gym and I'm like, what do I do? That's all I do. So anyway, whenever you go to a really good exercise program or whenever you buy one, because you can buy them and do them at home even now, those really good ones don't just tell you to get on the treadmill and run for hours. Have you ever noticed that? And if you go to a gym, you see those people. They get on the gym and they run for just like hours at a time and they do nothing else. And they're slim. I have my best friend, she's very, very thin, but she just says, I just can't tone up. I'm at the gym and I run on the treadmill for an hour, but I'm just still floppy. You need to do a little bit of this. And that is how we build the muscle. And building that muscle, toning it up, and burning the fat, that's the good balance. And then also with the diet, with the protein, in order to keep the muscles toned. That's kind of the overall, what a good workout plan should be. Okay. So here's your full sequence. This is a really good picture showing you what we start off with. When we start using the muscle, we said about 4 to 6 seconds. That's your stored ATP. And then we go into direct phosphorylation using the creatine phosphate and ADP, lasting about 10 seconds. Then we go into the anaerobic pathway for glycolysis. That lasts about 30 to 40 seconds. Say if you're sprinting, really short, quick types of exercise, and you're done, we'd stop there. But if we keep going for endurance types of activities long term for hours, and that could just be working in the yard, cleaning your house, whatever. It doesn't have to be running a marathon. If we keep using the muscle, that's where we go into the aerobic pathway. Because I think I mentioned before, if you really deep clean your house sometimes, you may wake up and you think, oh, why are my legs sore? And that's because you were working, you were moving and cleaning. It's not just exercise in the gym, it's just using the muscles. So we mentioned muscle fatigue, and there was a question about this also. Muscle fatigue is physiological inability for a muscle to contract. Physiological means that somewhere through that sequence of muscle contraction where we learn the physiology, there's something either lacking or we have too much of, that kind of thing. So it's an ionic imbalance usually. Maybe the levels of potassium, calcium, phosphate for the energy are interfering with the way that we can have EC coupling or the excitation-contraction coupling. Anytime we have prolonged periods of exercise, that could also damage your sarcoplasmic reticulum. It's not really good for you to use muscles all day long if you're not used to that, because it could damage the sarcoplasmic reticulum, which is where we're storing the calcium, and that calcium pours out and muscles start contracting. It's not good for your muscles. So again, it's messing with that calcium regulation. As far as complete lack of muscle contraction, that is from ATP being completely depleted from the muscles, and that's not usually something that we have happening on a regular basis. That would be in those extreme superpower situations where we use all of the muscle fibers. That would use a lot of ATP. So sometimes muscle fatigue, where we can't contract the muscle, that's where we don't have enough of something. But in other situations, we might get muscle cramps, and that's when the muscle stays contracted. And that is, like I said, maybe potassium, because potassium is where we relax the muscle. That's where we're trying to repolarize. And so if we're low for the potassium, where they say eat a banana if you're having muscle cramps, that's why that's happening. We're trying to repolarize with the potassium. So after exercise, using a muscle, in order for us to return to the pre-exercise state, 1, oxygen reserves, which is your stored oxygen, has to be replenished. We have to store back in with the myoglobin, the muscle globin. We also have to convert lactic acid to pyruvic acid, and sometimes that process doesn't happen very quickly because we have a lot of lactic acid, and like I said, that's when the muscles become sore. We also need to replace glycogen, which is one of our sugar sources where we made energy, and we also have ATP and creatine phosphate that we need to resynthesize and replace as well. So after we've used the muscle, there's a lot of chemical reactions that are still going on just to get back to the original stored and starting place. So we have this complete process called excess post-exercise oxygen consumption, where we're using an extra amount of oxygen in the process. So we've talked about how we can change the tension and the force of a contraction based on the strength of the stimulus. Now let's talk about strength of the stimulus and frequency of the stimulus. Now let's talk about the number of cross bridges and how that can change the force of your contraction. So the number of muscle fibers that we stimulate, more muscle fibers means a more forceful contraction. And also, one thing to note about your muscle fibers is that they all do not have the same diameter. Some of them are really huge diameter, meaning that they have a lot of filaments inside of them, a lot of actin and myosin, where some are smaller diameter with just a few myofilaments. The muscles in your hands, they have smaller diameter, whereas the muscles in your thighs, they have big diameters because we need a more forceful contraction there. We can also change the force of contraction based on the frequency of the stimulus, which we've already talked about. More frequency or higher frequency means a greater force. And we also need to consider the degree of stretch within the muscle. There's a limit where the actin and myosin need to overlap within the sarcomere. It's 80 to 120% of their normal resting length in order to generate the force. If a sarcomere is less than 80% overlapping, the filaments overlap too much and they can't pull and contract. Because remember, we pull the actin towards the middle and there needs to be some room. Also, if we stretch them out too far, myosin heads can't grab onto the actin and pull that way either. So there's a limit where we overlap, not too much, not too little. It's just right there where we need it to be. So in order to increase the force of contraction, which is, by the way, more force, tells you in this little picture, more force means we need to generate more cross bridges. If we have a larger number of muscle fibers that we recruit, that's larger motor units or more motor units, that's an increased force. Also, the size of the muscle fibers, you can see here's a little small one, here's some larger ones. And then the increased frequency of the stimulus, where we talked about wave summation and tetanus. And even the overlap, where we're stretching out or compressing down and changing the length of the sarcomere. So to give you a visual of how we can change the length to change the force of the contraction, we said that we want the sarcomere and the actin and the myosin to overlap around 80 to 120% of its resting length. And that's where we said myosin here in the middle, actin overlapping on either side. That's a good place so that we can slide the actin towards the M line, but then also not stretched out too far. So here, this is when actin is pulled completely to the M line. There's no way to contract that because actin is already pulled in all the way to the center. There's nothing for us to do. Over here, the sarcomere is so stretched out that the myosin heads can't grab on. They're not close enough to the actin to pull. So there is this number, that is where we generate a great force to the contraction, 80 to 100% of the original sarcomere length. For the velocity and duration of the contraction, velocity is speed, how fast the contraction happens, and then the duration is how long the contraction happens, or how long we stay in a contracted state. There's three things that can affect velocity and duration: muscle fiber type, load, which is the size of the object, and then also the recruitment, which is the number of muscle fibers. Muscle fiber type. Not all of your muscle fibers are the same, and we can classify them based on two characteristics. One is the speed of their contraction. We have slow fibers and we have fast fibers. And the speed of the contraction, whether they're slow or fast, is based on the speed at which we have myosin splitting apart the ATP so that we can have the energy to bind to the actin. And it's also based on the patterns of the electrical activities coming from the neurons. The second way that we classify muscle fibers is based on the metabolic pathway that they primarily use to make ATP. We have oxidative fibers and glycolytic fibers. Try to figure out which ones they match up to based on their name. Oxidative fibers means that we have to have oxygen in order to make most of our energy. And the only process that we said requires oxygen in order to make ATP is the aerobic pathway that's inside of the mitochondria. Glycolytic fibers, those are going to be the anaerobic pathways primarily, where we did not require oxygen, we used glucose in order to make the ATP. So in the muscle fibers, we have three different types of skeletal muscle fibers. We have slow oxidative, fast oxidative, and fast glycolytic. As far as the types of fibers that you have in your body, it depends on where the muscles are, and it also depends on your genetics. So, genes from your DNA determine what the types of fibers are that you have in your body. Some of us have more slow oxidative, some of us have more fast glycolytic. It just depends on your genes and also where the muscles are. The different types of fibers that we have in each of the muscles, where their position is based on the types of jobs that they're going to be doing. As far as slow oxidative fibers, they're great for low intensity and endurance activities. So that would be in locations where we're maintaining posture or long-term types of contractions. Fast oxidative fibers are medium intensity. So this would be sprinting, walking, so in the muscles of the legs. And then fast glycolytic, these are short-term, intense, and powerful contractions, like hitting a baseball or jumping, that kind of stuff. It's quick, powerful contractions. So to increase the velocity or the speed, we're going to use primarily fast glycolytic fibers, and we're also going to have a small load. Small object is what we're saying there. Smaller objects, we can increase the speed. We can pick them up very quickly and move them very quickly, as opposed to larger images or larger objects. We try to pick those up, it's a slow velocity because it's hard for us to pick those objects up. As far as the duration, which is the length of the contraction, again, the load, we can hold smaller objects for longer periods of time. The smaller the object, the longer we can hold that contracted state. And then we also have slow oxidative fibers used to increase the duration or the length of the contraction. So for table 9.2, it shows you a comparison between all three types of fibers. I want you to study the table and compare each of their characteristics. It tells you some of the metabolic characteristics, activities that they're best suited for, and it even tells you some structural characteristics. One thing that I want to point out is color. You may think that's kind of crazy. It says that slow oxidative, they're kind of a red color, fast oxidative, pinkish to red, and then fast glycolytic is a white pale type of muscle. That's kind of crazy because it's kind of like chicken. I told you I was going to ruin chicken for you. I don't mean to. But chicken, we have dark meat and we have white meat, right? And then sometimes if you carve the turkey, there's like that's in the middle. It's not really, really dark. It's not white. It's just kind of in the middle. And yeah, so you have the same thing. You have dark meat, you have white meat, and it's all based on the types of fibers. If you think about in the chicken, the white meat is in the breast meat, right? Where we don't really have long-term endurance, not where the chickens don't fly, right? So other birds fly, and they do have more dark meat, like quail. If you've ever eaten quail, they're all mostly dark meat, and that's because they do fly, and that breast meat is dark. But the legs, chickens walk around all the time pecking at the ground. They're dark meat, and that is because of the types of activity that they use those muscles for. So it's not exactly like that in your body, but it is very similar as far as the locations of where we have dark meat and white meat. As far as the load and recruitment, the load again is the size of the object. The greater the load, the larger the object, the shorter the duration or the time of the contraction that we can hold. The greater the load is also going to change the velocity of the contraction. The greater the load, the slower the contraction. Also, greater the load means we're going to recruit more motor units so that we can have a more forceful contraction and hold the load, hopefully, as long as we need to. But it depends on the size of the object. So this is just kind of showing you that we have different objects that we can, okay, so if we have a heavy load, we can only hold the contraction for a short period of time. Intermediate load is kind of in the middle. Light loads we can hold for longer periods of time. So we keep mentioning activities of exercise, and I just want to cover this, not just because it's in your book, but some of you are also interested. Some of you are more of the aerobic or endurance type of exercise, and then others are more of the anaerobic resistance type of exercise. For aerobic or endurance exercise, this is your jogging, your swimming, biking. Whenever you do those activities, not over a week of time, but this is over months of a time frame, you actually change your anatomy and change your physiology and the way that the muscles are working. You can grow new capillaries that supply the muscle. You can also increase the number of mitochondria so that we can supply more energy. We increase the myoglobin, which is what we stored extra oxygen with. So as a result, the more that you do these activities, the greater your endurance because we have 1, more mitochondria, 2, more capillaries to get the oxygen in there to use in order to make the ATP, and we also have more oxygen storage. So the longer we do these activities, not only do we get all of that to resist tiring and resist fatigue, but we also can convert the types of fibers. So if you're ever interested in running a marathon, they tell you you can start training and get your body ready, and you can actually convert fast glycolytic into fast oxidative fibers, which is where we're using the mitochondria and making ATP. Again, this is not something that happens overnight. But if you ever started exercising and you thought one day, okay, you know what, I'm going to start running, and you got out there, maybe in your genes, you were able to get out there and just take off, which some people do, but most people have to work at it. Whenever you run, you start off the first day and you feel like you're going to die. And you think, what did I get myself into? There's no way I can do this. I don't know how people do this. They're crazy. But if you keep doing it and you keep trying, then the easier it gets and the farther you can go because you're doing all of these things in your body. Resistance exercise, which is typically anaerobic, this is your weightlifting, isometric exercise. And this leads to muscle hypertrophy, which is where we increase the size of the muscles. So increasing the size of the muscle is not because we're increasing the number of muscle fibers. By the way, muscle fibers, the cells inside of these skeletal muscles, cannot divide through mitosis. The only way that we can make muscles larger is that we add more stuff into the muscles, like myofilaments. We can add more proteins in there, and that's why your muscles get bigger and they get stronger, because we have more myofilaments. More myofilaments means more cross bridges, that kind of stuff. We also make more mitochondria and store more glycogen, because glycogen is what we're getting through the anaerobic pathway, or using through the anaerobic pathway. But we also need to add in more connective tissues, like the epimysium, paramysium, endomysium, those packages, because the larger the muscle, the more package we need to hold the muscle together. So muscles must be active in order to stay healthy. If we don't use them, they atrophy, which means to degenerate, and they lose their mass. This could happen because we are immobilized, or because we have some type of nerve damage where we can't send the nervous stimulus to the muscle. And as far as how quick it happens, it's almost immediately. If you stop using muscles, they almost immediately start to atrophy and deteriorate. And that's something that you notice if you've ever been super, super fit and you go to the gym like every day and you're like, yes, I'm seeing results. And then you think, I'm just going to take a little bit of a break. It seems like it's within a couple weeks that it is so different. What happened? I promised I was in shape 2 weeks ago, but we lose it much faster than we can gain it. It takes a lot of work to get it back where we want it to. But the muscles, they do decline about 5% a day, which is very quick. You have to keep up with it if you want to stay super fit, if you want to stay super toned. It's something that you have to maintain all the time. All right, last but not least, we have smooth muscles. We're not going to have a whole lot of time to get into this, so I'll have to record and finish the lecture and post it online. But smooth muscle is not much of anything compared to what we talked about for skeletal muscle. Smooth muscle, as far as its location, remember, is found in the walls of most of your hollow organs, except for the heart, because the heart is where we have cardiac muscle. As far as the shape of the muscle fibers, and again, the muscle fibers, same thing for skeletal, it's going to be the muscle cells. They're very thin, they're short, especially compared to the skeletal muscles. They only have one nucleus that's located in the middle, and they're not going to have any striations. As far as the connective tissues, they don't have all three of the connective tissues that we've seen in skeletal muscle. They only have one of them, the endomysium, which is around your muscle fiber. Okay, all but the smallest blood vessels in your body, which are actually called capillaries, contain smooth muscles, and typically the smooth muscles in any of these hollow organs are going to be separated into sheets or layers. A longitudinal layer are where we have fibers running along the length of the organ, and then the circular layer is running around the circumference and the diameter of the organ. As far as what we do between the two, longitudinal changes the length because we run with the length, so that's where we shorten. And then the circular layer changes the diameter of the organ, and that's where we can make the lumen or the opening inside of these hollow organs smaller. We do see an alternating contraction between these two layers, and that's what we call peristalsis. Peristalsis is where we alternate between the two layers and contracting them, and that's used to either mix or even move materials through your body. So that's how we move food through the digestive system. It's how we move blood along the blood vessels, along with some of these contractions. There's a lot of places where we're doing this. So in the digestive system, this is probably the easiest place to identify the two layers. We have longitudinal layer, which is running along the length, and then the circular layer, which runs along the circumference. So if you look at it in this picture, that's where we can squeeze the fluids and squeeze the foods through the digestive system. It's kind of like this is gross, but it's kind of like those popsicles that you eat in the tubes. You know, you start at the bottom, you squeeze it through. Think about that being muscle contraction, squeezing that through, and that would be how your digestive system works. For the microscopic structure of smooth muscles do not have neuromuscular junctions, as we've learned about in the skeletal system. Instead, we have autonomic nerve fibers, and here's one I want you to also put a little asterisk next to, because we're going to learn what autonomic nerve fibers, right here, we're going to learn about what they are in Chapter 11. Autonomic nerve fibers, where they connect to the smooth muscle, they form these kind of enlarged areas, these little swollen areas that are what we call varicosities. In the varicosities, this is where we have our neurotransmitter. Sometimes it's acetylcholine, but other times it's something else, and we'll talk about those neurotransmitters later. Where we release the neurotransmitters, it's not called the neuromuscular junction. Instead, it's called diffuse junctions, because as we release the neurotransmitters, they are just released through diffusion and then attached to wherever they need to go. So here's one of our layers for the smooth muscle. Here's one of your autonomic nerve fibers. And the enlarged areas, those are your varicosities. You can see we have several. Inside of the varicosities, that's where we have the neurotransmitters, which would then be released at those diffuse junctions. So again, a smooth muscle does not have striations, and it's not going to have sarcomeres, myofibrils, or T-tubules. It's very simple compared to your skeletal muscle. Also, the sarcoplasmic reticulum is less developed, but it's still storing calcium. However, the sarcoplasmic reticulum is not the only source of calcium for your smooth muscle contraction. We get calcium from the sarcoplasmic reticulum and extracellular locations, which is outside of the cell. Outside of the cell, we have these little pouches that we call calveolae, and that's where we scoop in calcium from the outside of the cell, pull it in, and we use that for the muscle contraction. So if we look at these cells, this is one muscle fiber. Here's your nucleus right in the middle, and we have these little kind of white dots on the outside. Those are the calveolae, where we would scoop in the extra calcium. That's an extra source of calcium for the muscle contraction. Also, some ways that we're different from skeletal muscle. The thick filaments, we have fewer of them than we have in the skeletal muscle. And we also have fewer myosin heads throughout the length. So this means that we're not going to form as many cross bridges. Also, we do not have troponin for the regulatory protein. Instead, we have a regulatory protein called calmodulin. Calmodulin is what the calcium binds to. It's not troponin, calmodulin. The thick and the thin filaments, instead of running longitudinally, they actually run at a diagonal through the muscle fibers. And so whenever these muscle fibers are contracting, it's more of a corkscrew or a twisting type of contraction because of the direction of these myofilaments. Also, anchoring the filaments in place, we have intermediate filaments and something called dense bodies. These are responsible for anchoring all of the filaments and also resistance to tension, which is the pulling, and also, and that's kind of like the Z-disc in the skeletal muscle. So back to this picture, the lines that we have running in this direction, that's the intermediate filaments, and then the circles, those are going to be the dense bodies. All of your actin and myosin molecules would anchor to the intermediate filament so that whenever we contract and we shorten this muscle, it actually twists, and you can see the nucleus in the middle. It's twisting like a little corkscrew right in the middle. Okay, there is a table in your textbook. Make sure that you look at this table. It compares skeletal muscle, cardiac muscle to smooth muscle again, and this is the one that some of you were flipping through and trying to find in your textbook. Compares everything that we've talked about. Study it very, very. For the contraction of smooth muscle, the process is very slow and synchronized compared to skeletal muscle. The cells have gap junctions, which were the tunnels, the special types of junctions between cells that were tunnels that allow for electrical impulses to travel from one cell to the other. And those electrical impulses are going to be the charged particles like the sodium and the potassium that are stimulating the muscle contraction. Some of the cells in smooth muscle are self-excitatory, meaning they depolarize without an external stimulus. Because remember, smooth muscle does not require a stimulus from the nervous system, but the nervous system can modify the speed and the force of the contraction. So where we have these self-excitable cells stimulating the contraction sequence, we like to call these pacemaker sheets of cells, and they're there to communicate through the gap junctions and stimulate the contractions throughout the rest of the tissue. So again, neural stimulation and also some hormones or chemical stimulation can modify the rate and intensity of the smooth muscle contraction, but it's not necessary. The contraction of the smooth muscle is similar to skeletal muscle in a few different ways. One, that we are using the sliding filament mechanism where the myosin heads bind to the actin and we're sliding the actin over the myosin. Also, we've got to have calcium for the contraction. The calcium is not going to bind to troponin, but we will see that we have to have calcium, and there's going to be two sources for the calcium. That is from the sarcoplasmic reticulum again, and then also from the extracellular areas. We also need ATP in order to generate the sliding process, and as long as the calcium is binding to the regulatory protein, we have contraction. It's not until we remove the calcium that we relax the muscle. As far as how smooth muscle is different in its contraction sequence, number one, like we just said, the calcium is coming from two locations. It's not just coming from the sarcoplasmic reticulum, it's also coming from the extracellular space. And it's binding to the regulatory protein calmodulin and not troponin. As far as activating calmodulin, we have an enzyme called myosin kinase, and then eventually we're going to phosphorylate the myosin head with the myosin kinase. The phosphorylation of the myosin head had already happened before we started the process of contraction, whenever we bonded the calcium to the troponin for skeletal muscle. So what we're saying here is we have calcium binding to troponin, then we activate an enzyme which will then phosphorylate the myosin heads in order to form the cross bridge. We have to phosphorylate, which is very different from the skeletal muscle. In order to stop the sequence of smooth muscle, we require more steps than we saw in the skeletal muscle. We do have to take off the calcium from the calmodulin, and that's the regulatory protein, again, not troponin, and we also have to actively transport the calcium into the sarcoplasmic reticulum and the extracellular areas in order to relax the muscle. We also have to dephosphorylate the myosin, which means to remove the phosphate group that we use for energy, in order to stop the cross bridges. So for the sequence, you only have one picture to describe the sequence of smooth muscle. We said that calcium has to come from the extracellular and also the sarcoplasmic reticulum. That calcium binds to troponin, which was then the activated troponin, or not troponin, sorry. It's actually the calmodulin. Calcium binds to the calmodulin. That calmodulin then activates our kinase enzyme, and then the phosphorylation of the myosin head will then occur so that the myosin heads can bind to the actin molecules for the sliding. Again, there's not a troponin molecule. We're using calmodulin, which will then activate an enzyme for the phosphorylation of the myosin heads. At the point that we're ready to relax the smooth muscle, we have to remove the calcium from the calmodulin, replace them where they came from. We also have to remove the phosphates in order to detach your myosin from the cross bridges. Also from this picture, you know that there's not any nervous tissue, because smooth muscle does not require stimulus from the nervous system. We can have contraction all on its own. There's two types of smooth muscle. We have one that's called unitary smooth muscle, and it's commonly called visceral muscle. It's the one that's primarily found in all of the hollow organs except for the heart. So in these areas where we have these unitary smooth muscles, we have them arranging in the sheets that we've already described, the longitudinal and the circular layers, and we do innervate with the varicosities, where we have spontaneous action potentials happening in the smooth muscles, gap junctions, and we could respond to the chemical stimuli that we've already described. Multi-unit smooth muscle is going to be located in the lungs, in your larger arteries, erector pili muscle connecting to the hair follicles, the iris of the eye. There's fewer gap junctions here, and they actually depolarize spontaneously, and the spontaneous depolarization is rare. One way that it is similar to the skeletal muscle is that it has individual fibers that contract independently, and so we can have a more forceful or a more less forceful type of contraction. So we get what we call graded contractions, different strengths of contractions. So for a couple of homeostatic imbalances, one is called muscular dystrophy. This is a group of inherited muscular destroying diseases that usually appear around childhood. And this is when muscles enlarge as a result of fatty connective tissue being deposited around the muscles. After these deposits, they atrophy or degenerate and weaken. Duchenne muscular dystrophy is the most common and also the most severe type of muscular dystrophy, and it's caused by a defective gene that is forming dystrophin, which is one of those anchoring proteins. It's inherited through sex-linked traits, and that's male or female. It's carried by the female and expressed in males. So the dystrophin is one of the cytoplasmic proteins that links all of the cytoskeletal elements to the extracellular matrix. And it's basically there to stabilize the sarcolemma. So the sarcolemma is going to become very fragile in these muscular dystrophy situations, and they tear very easily, and that causes an excess entry of calcium, which leads to damaged contractile fibers. We get a lot of inflammatory cells that are coming into the area and actually destroying the muscle tissue, and muscle mass will decline. Victims, as far as what they usually start out for their symptoms, they start off very clumsy and they start falling, and we start to notice that around the ages of 2 or between the ages of 2 and 7. Currently, there's no cure for muscular dystrophy, but we can prescribe what's called prednisone in order to improve the muscular strength and function. The myoblast transfer therapy has been kind of disappointing because we're trying to implant stem cells, not really having much success with that. But there are some other types of therapies like viral gene therapy, stem cell therapies that are showing more promise. As far as the fate of the individual, again, there's no treatment here, so usually the respiratory system starts to fail, and it says that typically patients die in their early 20s. That's not all the time, but they usually do have an early death.